Hi, my name is Bailey McLean, and we're going to go over synapses of chromosomes. So the type pairing of homologous chromosomes is called synapsis. It takes place in either prophase 1 or 2 of meiosis, and if you recall, meiosis is the process of producing viable gametes with the intent to reproduce. So synapsis supports the exchange of chromosomal segments between non-sister homologous chromatids. So viability, it's really important for meiosis. Gametes are viable if you have a centromere, all loci are present, and there's no duplication. So I like to think of it as can, or like using the mnemonic can to remember if the gamete is actually viable. So we're going to look at two individuals, and the chromosomes for this one is going to be MLI, centromere, JKHG, and GHI, centromere, JKLM. So as you go through this image, you can look at the different colors and it will show you the different gametes. You can see 2 of the 4 are viable, while 2 of the 4 aren't. And you can see the brown color and the gray color are not viable because they do not have all the loci present. There's no duplications and they do have a centromere, but because they don't have all the loci, they're not viable. And then for individual 2, we have MLI, centromere JKHG, and GHKJ, centromere ILM. So to get more like matching, we flipped this chromosome and it shows that they're identical. So because they're identical, all 4 gametes are viable. There's no crossing over, and this means this individual has a higher level of fitness because all 4 gametes are viable. And here's my citations. Thank you.